Hello everyone, and this is Supriya Gupta, and I have been teaching the series View Under the Microscope, that is the Histology series for first year students. And I have made 3 videos till now, and if you haven't checked them, go and check. Okay, and today's topic will be continuation of the connective tissue that we have been studying in the third video. And today we will be discussing about the cartilage, the bone, the muscle, the nervous tissue. Okay, and I'm really sorry for this sun-kissed appearance because I don't have any electricity at my house. Sorry. So, a very quick review. Okay, this PPT will be containing the histo of the cartilage, the bone, the nervous tissue. Okay, now, first of all, let's go on with the cartilage. So, for an overview, what is a cartilage? You know, cartilage is a connective tissue, and it is basically the skeletal basis of some part of the body, and it is firm, but it is not rigid. And difference from the bone, if you see, matlab, consistency wise, so it will be flexible and it is elastic. The bone is not. And now, second, components of the cartilage. Okay, so cartilage is not a cartilage is not a single cell. Okay, it is a connective tissue. So contain, if it is a connective tissue, then it must be having some components. So for components, first of all, we have cartilage cells. Cartilage cells are known as chondrocytes. Okay, and they lie in the lacuna. Lacuna means a space which is present in the matrix. And first, these cells are very metabolically active, and they have euchromatic nuclei. And metabolically active, the cells have prominent endoplasmic reticulum, the mitochondria, and the Golgi apparatus. And but as they grow, the nuclei become heterochromatic, and the organelles become very less prominent. Now, coming to the ground substance. Ground substance is same like a protein and carbohydrate, and the most important carbohydrate here is glycosaminoglycans. Okay, and this will include chondroitin sulfate, keratin sulfate, and hyaluronic acid. And these are very important questions you see here. These are the very important questions, that what is the ground substance of the cartilage tissue. Okay, now uh, we have seen the cells, we have seen the ground substance, and the last will be the fibers. Fiber will be collagen fibers. Which collagen fiber? Normally cartilage contain type 2 collagen fibers, but there are some exceptions like fibrocartilage and the perichondrium, which contain type 1 collagen fibers. Okay, so types of cartilage. In a adult human body, we have 3 types of cartilage: the hyaline cartilage, the elastic cartilage, and the fibrocartilage. Okay, now first of all, let's see what is a hyaline cartilage. Hyaline cartilage are basically a type of cartilage in which an isogenous group of chondrocytes, they lie in a nests. Okay, the chondrocytes are separated by a homogeneous basophilic matrix, and chondrocyte will increase in the size from the periphery to the center. Okay, now you know what is perichondrium? Near the surface, the cartilage cells become flattened and they merge with the overlying connective tissue, and this combination is known as perichondrium. The perichondrium is further divided into the outer fibrous layer, the inner cellular layer. An example where the hyaline cartilage is present is articular surface of the bones. Okay, where the bones form articulation, the joints. Okay, so the articular surface of the bones. Second is the end of ribs. Then the respi passage, that is the nose, the larynx, the trachea, the bronchi. Okay, now coming to, see, where should I keep it? Okay, so in hyaline cartilage, the you will see the structure like this. First of all, can you see this structure, this length, this breadth? This is the perichondrium. Okay, and these spaces are basically known as lacuna, in which the chondrocytes are present, and this cell is actually the chondrocyte, and these are the isogenic group, and they will be embedded in the extracellular matrix. So this, the whole the background material is basically the extracellular matrix. So come here in the real picture. Can you see here? This is the perichondrium, which is not here in the picture, but because the flattened cells are there, hence it is marked here as perichondrium. And these white spaces are basically the lacuna, and inside the white spaces, can you see the nuclei of the cells? They are basically not the nuclei, they are the chondrocytes. Okay. Now, this is the picture you will have to draw in your copy. Okay, you can see the chondrocytes, you can see the nests, cell nets, the group of the cell. These are the nests which contain the isogenous cells. Okay, and perichondrium here, the last is perichondrium. Okay. Now coming to the elastic cartilage. So elastic cartilage, here the characteristic is this कि chondrocyte will be within the lacuna and but they will be surrounded by bundles of elastic fibers. You see elastic cartilage, elastic fibers. Okay, and here also the perichondrium will be present, and distribution means the examples, that is pinna. You see this pinna. Now, the lateral part of the auditory tube means the eustachian tube, ka lateral part, and then we have the cartilages of the larynx, that is the epiglottis, the corniculate cartilage, the cuneiform. Okay. Now, this is how the elastic cartilage looks like. You will see the perichondrium here in the above, first of all, and in the perichondrium you can see fibroblast here. Yes, and these are the lacuna, the white portion, and inside the white portion there is this densely colored chondroblasts or chondrocytes. And see, in the ground substance, in the previous slide, the ground substance did not had any elastic fiber. Here the elastic fibers are present in the ground substance. Yes, and see how densely these stains, how densely, because these are basically the staining which is taken by the elastic fibers. Okay, see, this is how you will make it, and you will just have to add the elastic fibers. Now coming to the fibrocartilage, this is the third type of cartilage in our body, is fibrocartilage. And here there is also prominent collagen fibers arranged in the bundles with rows of chondrocytes intervening between them. Okay, and here the perichondrium will be absent, and it is actually confused with a tendon, because you know the chondrocytes are rounded here, but the tendon fibroblasts are flattened and elongated. Hence, fibroblasts, see, there is no perichondrium first of all. The second will be the chondrocytes are present in rows, and the isogenic groups are also present. And here there is not elastic fiber, here is collagen fiber. And see, this is how it looks like. The lacuna, inside it the chondrocytes, and these are the bundles of collagen fiber. Yeah, you remember the video that I showed you in the previous connective tissue slide? I showed you the collagen fibers, how they look like. So this is how they look like. Okay, see how beautiful this picture is. You see the bundles of collagen fiber is there, and these are the rows of chondrocytes. And the example, that is the intervertebral disc. Okay, this is how you will make it, the rows and the collagen fibers. Now uh, very quickly coming to the bones. So coming to the bones, first of all, bones overview. These are the rigid form of the connective tissue, okay, in which the extracellular matrix will be impregnated with the inorganic salts. And which salt? The very important salt is actually calcium hydroxyapatite. Okay, here in the simple terms, I have written calcium phosphate and carbonate, which will provide hardness to the bone. Okay. So, components of bone tissue, here also we will have components, cells, fibers, and ground substance. So, cells, there are many types of cells in the bone. So, first of all, the osteoprogenitor cells. Osteoprogenitor cells have a mesenchymal origin, and they actually proliferate to form the osteoblast cells, and they will resemble fibroblast in appearance. Now coming to the osteoblast. Osteoblasts are the main bone forming cells which are derived from the osteoprogenitor cells, and line growing surfaces of the bone sometimes give an epithelium like appearance. Okay, and these cells are very numerous shaped and have gaps between them. The nucleus will be ovoid and euchromatic, and the cytoplasm is very basophilic due to many rough endoplasmic reticulum. And their function is to lay the organic matrix of bone, which will be including the collagen fibers and calcification of the matrix. Okay, now coming to osteocytes. The osteocytes are the cells of the mature um, um, osteoblast. Okay, these are the mature osteoblasts which have been differentiated. Now they will lie in the lacuna of the bones, and they represent osteoblasts that have become imprisoned, means they cannot go anywhere else now, in the matrix during the bone formation. They have cytoplasmic processes arising from the osteocyte to establish a contact with the other osteocytes, and bone lining present on the surface of the bone. They have eosinophilic cytoplasm because during the differentiation the endoplasmic reticulum decreases, and hence they become eosinophilic, and they will maintain the calcification of the bones. Now coming to the osteoclast cell. See, in our body if कुछ अच्छा है तो कुछ बुरा भी होना जरूरी है. जैसे कि osteoclast is not always bad, osteoclast is very important. नहीं तो हमारा bone का remodeling नहीं होगा, और अगर bone का remodeling नहीं होगा then our bone will be weak. Okay, so this is a physiological cell. Okay, remember this is a physiological cell, physiologically active. तो osteoclast cell is basically a bone removing cells. Bone removing means, अगर osteoblast को भी over action करता है then it will form many extra bone. तो osteoclast are the cell which will remove those extra part, and they will remodel the bone into good figure and shape. Okay, now they will be found in relation to the surface where the bones will be removed, and they occur in the pits called reabsorption bay or the lacuna of Hosch's. Now they are large and they are multinucleated. They are just giant cells. Giant cells are the multinucleated cells. They are basically those giant cells, and they show ruffled borders, and many Golgi apparatus in mitochondria and lysosomes are present, and they have acidophilic cytoplasm. Now coming to the pictures. See, this is how the osteoblast looks like. These are the cytoplasmic extension but not the cytoplasmic process. Okay, this is how an osteocyte looks like. Osteocytes, I told you now, these are the cytoplasmic projections that I was talking about, cytoplasmic strands to maintain the connection. And these are the osteoclast cells. Osteoclast cell, you see, there's so many of nuclei, there's the multinucleated giant cells, and these are the ruffled borders, you see, and this is the cell surface where the osteoclast is actually acting and remodeling the bone. Now the cells we have done. Now the bone tissue. The bone tissue present, the fibers in the bone tissue is basically type 1 collagen, and the ground substance is basically glycosaminoglycans, proteoglycans, and then we have many other inorganic things like calcium phosphate and magnesium carbonate, hydroxyl ions, chloride, fluoride, citrate, sodium, potassium. Okay, now types of bone. Types of bone can the bone can be divided on various subtypes based on histology, maturity, and the manner of development and the shape of bone. So first of all, on the histology, the bone can be divided into compact bone and cancellous bone. Okay, based on the maturity, the bone can be divided into mature or lamellar bone या फिर immature or woven bone. Manner of development, they can be developed from cartilage, they can be membranous in origin, they can be membranocartilage in origin. Okay, now shape of the bone can be long bone, short bone, flat bone, or irregular bone. Now here we will be studying histology of the compact and the spongy bone only. Okay, and other classifications are basically not the part of the histology, and they are the main general anatomy part. Okay, so the compact bone is made up of concentric lamella, and these lamella will contain lacuna in which you will find osteocytes and canaliculi, and these concentric rings surround a cavity which is known as Haversian canal, and these Haversian canal will give way to the blood vessels, and with the lamella around will constitute the Haversian system or a osteon. Okay, osteon is the single unit of the compact bone. Now, between the osteon there are interstitial lamellae. Near the surface of the compact bone, the lamella are parallel to the surface, and these are known as circumferential lamellae. Now, these Haversian canal actually communicates with the marrow cavity and external surface of the bone through another canal which is known as Volkmann's canal. So this is actually, this will be clear when I show you the picture. See, this is a osteon. You will see here, first of all, you see it is basically a cross section. Okay, the bone has been cut like this. This is my, if this is a bone, you will cut the bone like this, and this is the overlay you will see. Okay, so first of all, this part, this part is actually the compact part, and this part is actually the pore which have pores. Na, this are the spongy part. Okay, so if these channels are containing the vessels, then this will be Haversian canal, and these are the perforating channels. Now, the bone has the overlying covering which is known as periosteum, and if I show you in the next picture, this is how the bone looks like actually. This is the Haversian canal in which the vessels will run. These are the concentric lamella, and in the lamella contains the lacuna in which the osteocytes will be present. And see, every osteocyte will give so much of cytoplasmic processes to communicate with the other osteocyte. And this is the Volkmann's canal. See, this is the picture you will draw in the histocopy, and try to draw it in black color only. Now coming to cancellous bone. So in cancellous bone, we have meshwork of bony plates or rods called trabeculae. Okay, you see in cancellous bone you have many spaces there, and made of many lamella between which there are lacuna containing the osteocytes and the naliculi, and the trabecula actually encloses a wide space which is filled by marrow, bone marrow, and these are also known as spongy bone. And here the most important thing is the Haversian system is actually absent here. Okay, now here you will see the picture. The compact bone and the spongy bone, you see this is the spongy part, this is the compact part. Okay, the overlying thing is periosteum, and these are the central canals, Haversian canals. Okay, and this is basically a osteon. Here also you can see osteon will contain numerous lamellae, the canaliculi that are connecting to one another, the lamellas, and these lamellae will contain lacuna which will be containing the osteocytes. And the trabeculated part is actually the trabecular bone, that is a spongy bone, and these are the Haversian canals, and the connecting part is the Volkmann's canal. Wherever the Haversian canal joins, either it is opening in the surface or it is opening in the marrow cavity, it will be opened by the Volkmann's canal. Okay, same here, you can see one osteon is there, the compact bone, the spongy bone, the cancellous bone. This is the Haversian canal, and this is the Volkmann's canal which is connecting the Haversian canal. Yes. Okay, so this is how a spongy bone looks like. This is the ribbon-like part, you see? This is the ribbon-like part. This is actually the bone, and this is the spongy bone. This is how you will make it. You see? This is the spongy bone basically. This part, and we have bone marrow, we have trabeculae, we have fat cells, we have nucleus, everything. So this is the marrow spaces. Okay, this is the marrow spaces, and this is the endosteum, and this is spongy bone. This is spongy bone, the lacuna and the osteocytes inside it, and this is the red bone marrow. Okay, now now Coming to the muscular tissue. So, muscle tissue is predominantly cells that are specialized to shorten in the length by contraction that results in movement. These are the specialized tissue which will contract and will cause movement. Okay. So, types of muscular tissue. You know, there are three types of muscular tissue: the skeletal variety, the cardiac, and the smooth. Skeletal muscle. Uh, the skeletal muscles are those voluntary muscles that we, by our own will, we can move it. Like, see, this is the biceps, and this is the skeletal muscle, and we can move our hand because we can move the biceps by our own will. Okay, these fibers are actually multinucleated with peripheral nuclei and multiple nuclei due to the fusion of the mesenchymal myoblast during the embryonic development, hence forming syncytium. Okay, each muscle fiber is actually composed of myofibril or myofilament, and the last portion will be actin and myosin, that is the smallest unit. If we are not taking the A and I bands, of course, and they form crisscross pattern. These are the smallest unit which is actually causing the contraction. Now, muscle is surrounded by the connective tissue which is known as epimysium. Each muscle is surrounded by epimysium. Each muscle fiber are surrounded by endomysium. Okay, and these are under conscious control, that is the our own voluntary control. And neuromuscular spindles are the specialized stretch receptor in almost all the skeletal muscles. You know, neuromuscular junction and all. Okay, in lateral section, the fibers will show transverse striations. The fibers will be long, they will be parallel, and most important thing which will differentiate it from the cardiac tissue is that it is without branching. There will be no branching, there will be only parallel fibers. Okay, many flat nuclei will be present in the periphery, and they will be fibers will be separated by connective tissue. Now, each fiber will be surrounded by endomysium. Now this fiber and this fiber, these two will be surrounded by the perimysium, and now and forming fascicle. Fascicles will be surrounded by the epimysium. Okay, see here, these are the single muscle, sorry, okay. Okay, this is the bone, this is the tendon, this is the muscle belly. Okay, muscle belly. You see here so many of fibers, so many of fascicles and all. So the very good picture. Where is the good picture? Can't find my picture that I added. Picture has gone blurred actually, and oh, sorry. Yes. So you see, this is a single muscle fiber, and this is actually surrounded by endomysium. Many of these muscle fiber will form a bundles, that is will be a fascicle. This fascicle will be surrounded by perimysium, and many of these fascicle will actually form the muscle belly, and this muscle belly will be surrounded by epimysium, also known as deep fascia. Okay, so if we go in the microscopic section, then you will also, each muscle fiber will be further having many of the bundles of the myofibril, and each myofibril, you know, we have I band, A band, that is a dark and light bands. Okay, so now here, this is how the skeletal muscle will look like. You see, this is a longitudinal cells. These are the longitudinal cells with no branching whatsoever, and the nuclei is so peripheral in location. These are the peripheral location. And you can see also the banding pattern, you know, dark and light bands. So beautiful picture is there. Dark and light band, but they are parallel to each other, and hence they are striated muscle. Yep. So Here also you will see striations are present, but there is no branching. So this is actually an explanation of this photo. These are the nuclei. There are multiple nuclei in the same muscle fiber, and hence they are known as syncytium. They have striations because of the light A band and I band, and they have connective tissue in between them. See, this is the connective tissue. Okay, this is how you will make it. Now coming to the cardiac tissue, cardiac muscle. So cardiac muscle, as name suggests, cardiac muscle is present in the heart and the large vessels which are attached to the heart. Now the cross striations of the actin and myosin are very similar as in the skeletal muscle, but here now you will see we have intercalated disc that contain gap junction, and this gap junction is the main reason that our cardiac muscle has the property of autorhythmicity and spontaneously generating stimuli. And these fibers are made up of cells which have a centrally placed nuclei. There the nuclei was peripherally placed, here is the nuclei centrally placed, and the striations will be present because of the A band and I band, and you will see a perinuclear halo also around the nucleus, and the adjacent cells will be separated by the transverse intercalated disc. And the most important thing here that the fibers will show branching. There the fibers were parallel, but here the fibers will be showing the branching pattern, and here the blood vessels will be seen also. Okay, so this is how, see, these are the branching. The so many branchings are there, and you see these are the intercalated disc. These are the intercalated disc, the places where the branching is occurring. We are having an intercalated disc, and this is the cell, muscle cell nucleus. It is not syncytium here. Okay, it is one cell is containing only one nucleus. There the one cell was containing multiple nucleus, and hence multiple nuclei, and hence that was a syncytium, but here it is not a syncytium. They are acting as a unit because of the intercalated disc which are containing the gap junctions. Okay, now there is a capillary, and see here also so many branchings are there, so no problem in identifying the both. Okay, this is a branching. This is how, this is so much of branching pattern you can see here, can't you? Yes, these are the intercalated disc we have. These are the जो dark आपको दिख रहा है ना, ये transverse these are the intercalated disc, and this is the nucleus, and around the nucleus this is the perinuclear halo is actually of the glycogen. Okay, here is the halo. Okay, now coming to smooth muscles. Smooth muscles are found in the hollow organs and blood vessels, and these muscles are actually bound by zona adherens, and also they have gap junction, and they also contains actin and myosin, but without any crisscross striations. You cannot see striations in the smooth muscles, and the fibers are very, from the NCRT book, the fibers of the smooth muscles are fusiform in the shape, and they have single and centrally placed nuclei. So this is a smooth muscle, you see this picture. Yep. So this is a smooth muscle. One smooth muscle, two smooth muscle, three smooth muscle. This is a smooth muscle, spindle shape, and one centrally placed nuclei. This is how you will draw it. Okay. And there was no striation. Okay. Remember that. And in LS we will see spindle shaped cells with tapering ends and the centrally placed nuclei which is elongated, and there is no striations. In transverse section the spindles will be cut in different places, and hence the nuclei will be seen in those cells which are cut through the center. Okay, because when we will come in the other cross sections of the tissues, you will see the the shape of the smooth muscle will change because the sections will change in which they are cut. This is a spindle shape, you see. These are the longitudinal section, and this is actually this is actually not the transverse section, I guess, but the way these smooth muscles are arranged, I guess this is the transverse section. Okay, so these are the two different sections of the smooth muscle. So coming to the last part, that is the nervous tissue. So coming to the nervous tissue, you know, there are specialized tissue in our body to conduct the impulses from one body part to the another body part, and these cells are actually the functional units of the nervous system, and they are known as neurons within the brain and spinal cord. The neurons are actually supported by special type of connective tissue known as neuroglia. So the coming to the structure of the neuron, you know better than me because you have studied in CRT right now. So I won't go in deep. So you have a cell body or the perikaryon, and then we have dendrites, and then we have axons, then we have synapse with the another neuron, and in relation to an effector organ, we have muscle or gland where the nerves is actually going to supply. Either it is taking the sensory sensation away from it, or it is giving a motor sensation to the gland or the muscle. Okay, so yeah, this is how the neuron actually looks like under the microscope, and you will see the nucleus, you will have the cell body, you will have the dendrites, and you will have the axon part, and this is how you will draw it. Okay, the drawing is not clear actually, I guess. Okay, I will provide the drawing in the, if anyone needs it, just message it in the MME WhatsApp group. I will send it. Okay, so what would I say? The drawing is actually very distorted now. These are the dendrites. These are the Nissels granules inside it, the blue parts. This is the nucleus, and this part is the axon hillock. Axon hillock is the most sensitive part in a neuron. Then we have the axon. We have, this is the lamellated, okay, neurilemma, or the Schwann cell, you can say, and this is how they are synapsing, and this is the motor end plate, you can say. Okay, and these are actually the neuroglia cells. Okay, these are the supporting matrix cells. These are the neuroglia cells where the neuron is actually placed to support it. So this is the cytoplasm, this is the nucleus, this is the nucleolus, this is the microglia, this is the perikaryon, the cell body. The Nissels granules are actually the rough endoplasmic reticulum, and this is the axon hillock. Okay, this is also the nervous tissue. Coming to types of neurons on the basis of the number of processes. Of course, you will have, if only one axon is present, then it is unipolar neuron. Two processes are present, then it is bipolar neuron. Many processes is multipolar neuron. The processes here can be the dendrites or the axons, both. Okay, both are the processes of the neuron. Now, on the basis of the function, they can be sensory neuron or they can be motor neuron. With the motor neuron, the impulses will be coming from the CNS up to the effector organ. If it is sensory, then the impulses will be carried on from the effector, from the afferent path to the CNS. Okay? And basis of the myelin sheath, we have myelinated and we have unmyelinated nerves. Okay, so these are the types of neuron. You see, different types of neuron I have been seeing, showing you. These are the olfactory neuron, retinal neuron. These are bipolar neurons. In unipolar neuron, we have touch and pain sensory neurons. The anaxonic is the amacrine cells. The amacrine cells in this present in the eye, the layer of the retina. Okay, and they have Purkinje cells. We have pyramidal cells. We have motor neurons. These are all multipolar neurons. Example. Okay, if you draw like this, I don't know why the diagrams have been distorted here. Okay, so the basic structure of a peripheral nerve, you will see these are all surrounded by a layer of endoneurium. The nerve fiber will be surrounded by the layer of endoneurium. Now, these endoneurium will contain collagen, fibroblast, Schwann cells, endothelium cells, and macrophages. When many of the nerve fiber together forms a bundle, they will called as fasciculi, and they will be surrounded by perineurium. and this perineurium is actually made up of the layer of flattened cells which are separated by the layer of collagen fiber, and these fasciculi will be held together by epineurium. You see, this is same like the muscle part. Yeah. So first of all, there will be a neuron. The neuron will be having an axon. This axon will go, and this is actually the endoneurium. This endoneurium is forming a fascicle. Fascicle is actually surrounding by the perineurium, and there are many fascicles with artery, vein, fat, and the whole thing will be surrounded by the epineurium. Okay. So this is the diagram of a peripheral neuron, and this is how you will see it in the microscope view. Okay, so I know this video was a bit very clumsy, but sorry, and thank you, and see you again in the next video. And I will be very grateful if you can add some comments below down there so that I can improve my video. So till then, take care. Bye.